Library.
All right, what we want to do now is bring the speakers to the front of the room here in Washington, DC, and Dr. Lavery, we will have you here virtually. And at this point, we have about 30 minutes set aside from here for questions from the committee to the presenters. So let's start with Dr. Kabenick. Yes.
Well, first off, I thought that was an absolutely fabulous panel, all five speakers. And Kevin, I want to especially thank you for being so forthcoming about your work and sort of beginning to drive this conversation. Your comment, there's no societal precedent for this kind of work. I found myself wondering how many societal precedents there are for what you were doing, actually. There are some, but it's remarkable to have scientists telling us at the point that we're thinking about the ethical ramifications of their work, this is unprecedented. Usually what we hear is, this is very much like other things that have been happening. So I have two questions. Actually, they're for you, Kevin, mainly, but others can jump in. And Ken, you answered this a little bit, but I'm especially interested in Kevin's perspective. First, we're charged with thinking about gene drives very generally. It's not pegged to a particular technology, but some of the recommendations you have, I wonder if they are pegged specifically to the CRISPR-Cas9, like the reversing and immunizing drives. Is that even feasible with homing endonucleases? And as sort of a follow-on to that, is there any likelihood, what do you think is the likelihood that CRISPR-Cas9 is going to be superseded by something else in the next, I don't know, 5 years or so? Can we, what hope can we have of coming up with recommendations that will be applicable to the next wave? Maybe I should stop there, that's good.
All right, so those are very good questions. Now, with respect to, I'm going to address the homing endonucleases one first and the generality. So Austin Bird has done a lot of work on homing endonucleases and reversibility. The way that the homing endonucleases can be reversed is specific to the application. And in particular, the application that they're working on for Anopheles gambiae is a suppression drive that targets sequences on the Anopheles gambiae X chromosome, shredding it during meiosis, thereby biasing the population towards males. That can be reversed. So if that is placed on the Y chromosome, then it becomes a driving Y that is favored because it outcompetes the X. It ensures that the Y is passed on to all surviving progeny. It can be reversed by simply creating the resistant version of the X chromosome and releasing it. Since the goal is to suppress the population, if you release a resistant version, the resistant version will rapidly take over, and you will have, in effect, reversed it. Similarly, for any highly deleterious change that you're making, it can be reversed in that manner. Otherwise, not necessarily. But homing endonucleases are so hard to retarget that it's hard to imagine what else you're going to do. That's really the one application because the site is already there and there's already a homing endonuclease that targets it. More generally, with respect to gene drives, I would say that yes, my recommendations were very specific to CRISPR RNA-guided gene drives in particular. And there are other means of doing threshold-independent gene drives. They are not unlikely to be as evolutionarily stable or as versatile in general. And I would argue that the considerations are very different for threshold-dependent drives, wherein you don't have the same sort of risk, at least at the laboratory level. But as soon as you're talking about field releases, some of the threshold-dependent gene drives are arguably better approaches because you can control the spread. That is, you could confine the genome change that you are attempting to drive to, say, an island population of invasive species with no risk of having it spread to the mainland. And so one of the most powerful approaches in the long run might be using an approach like underdominance to change the sequences of the target population on an island without risk of that spreading to the mainland, and then following that up with a CRISPR RNA-guided gene drive that targets the new sequences only, and therefore would not affect the native population wherever it came from. So there's going to be, you are going to have to address some of the threshold-dependent drives as well. But our recommendations are really for the threshold-independent ones, because those are the ones where there is a risk that our laboratory experiments specifically could end up impacting the lives of others.
Let me add one small point to what Kevin was suggesting. Not only do I like the idea of an island, but I also like the idea of making the alterations to very small niche species with an immunization drive and a reversal drive in your back pocket. It's belts and suspenders and other precautions, and the reason is that as we're learning more about the processes and their stability over time, I kind of want lots and lots of measures in place.
And to your second point about something superseding CRISPR, you never know. Nature has amazing treasures if we only know how to look. That said, CRISPR is pretty darn easy to use. So something even easier is really hard to imagine. It's just express the protein, express the guide RNA, matching the sequence that you want to cut. It really is something that literally high schoolers are using now. So it could happen, but you never know.
Okay, Dr. Delcourt, follow up on this?
Yeah, I just wanted to ask on the question of threshold independent and dependent gene drives, how confident are we to distinguish between one and the other? And have you considered the possibility of limiting research to threshold dependent gene drives for a certain amount of time before we would do experiments on threshold independent gene drives?
So the problem with threshold dependent is that you have to, for the evolutionarily stable ones, in general, you have to release sufficient of whatever the organism is to comprise more than 50% of the population, which in the case of something like you want to address malaria is just not feasible. There are going to be cases where, I mean, in general, if you can use a threshold dependent, then great, by all means. But the scale limitation is a pretty severe one.
Doctor, follow up on this one? Dr. Hayes?
I have one question. Presumably, there's no evidence of Cas9 crossing species barriers because, by definition, they don't interbreed to produce fertile offspring. Is that correct?
So there is Cas9 currently in the bacteria in my gut and in your gut and in all of us. It's actually, there are CRISPR systems found in roughly, I think the latest figure is 46% of bacteria and somewhere around 90% of archaea. Not all of those are Cas9 specifically, because that's a type 2 CRISPR system and there's 3 types. But it's tremendously abundant in the biosphere. That's another reason why attempting to simply control access, not only does every molecular biology laboratory have one, but you can pull one out of a fecal sample at any point. It's just that abundant. That said, it's not in any metazoan. In fact, it's not in any eukaryote that we know of. The only examples of Cas9 being present in a eukaryote are when we have put it there. But that's not to say that it's not readily available. And if you simply run a PCR on a blood sample, odds are you could probably pick up Cas9 DNA fragments. So the notion that it's not available at all, given the ubiquity of horizontal gene transfer, I would say it's out there. Now, being out there and available to a eukaryotic organism, we know that it's really useful. You can do transcriptional regulation, all sorts of things. Maybe it would be advantageous for them to have it, and evolution would eventually repurpose it. And by making it available, not just to an isolated case, but fixing it in an entire species, maybe that would change the evolutionary trajectory of that species. But then that's something where you're looking at 10,000 years down the line. Very, very speculative.
All right, so there's no evidence of putting it into a new species and that getting transferred to a predator or anything like that so far?
So that's very common with other types of gene drives, in particular transposons. So a quarter of the cow genome originated in snakes, from a transposon in snakes that was probably transmitted by a tick into the ancestor of the cow, and since replicated until it was a quarter of the cow genome. So that sort of thing happens all the time. But Cas9, because it's so site-specific, that is, it needs a guide RNA targeting a particular sequence, and the odds of, and the sequence is long enough that it's very unlikely to happen by chance. That's why it's so useful to us, because we can target a particular site in the genome with very little risk that it's going to target any other site. It's a 20 base pair recognition site sequence, typically, and 18 bases is usually sufficient to be unique in a human genome.
The note here is that if there is a degradation or fall off in efficiency, it also reduces the power of the gene drive to go through the wild population.
And you said it was out there. Does that not confound your ability to detect it it's released.
Great point. It is out there. The odds that the particular version that we most commonly use in the laboratory is going to be out there, because there's 2,500 orthologs of Cas9 that we've characterized so far. Also, the structure of the guide RNA that we use is a fusion of two RNAs. So the natural version has two separate ones, separate pieces that associate, and we typically fuse them together, and that makes it more efficient, at least in metazoans and plants. So in that sense, you would be able to pick it up. Could you necessarily pick it up by PCR? No, because we can vary the guide sequence and they could use a slightly different Cas9 and so on. But if you pick up an instance of the Cas9 gene of any sort associated with a single guide RNA fused, that's obviously human in origin. And if you even find the natural version, because you could probably build a gene drive with the natural version, it might not work as well, but you probably could. You could look at what sequence it targeted and say, is that a sequence in this organism of interest? And that would tell you.
Okay, Dr. Heitman.
I'd like to go back to the question that I asked the sponsors, the sponsor representatives. When we were talking early about whether the tone of the scope suggested that this was inevitable, that it would always move forward, Todd, you made the comment obliquely that maybe you didn't think it was. Do you envision places where there ought to be a hard stop? And this is, I guess, for everybody, including Dr. Lavery on the phone. Is there an inevitability in your future view that we need to be attentive to as many, if not, well, as many as we can, of the possible permutations of this technology? Or do you think we need to also focus on some just won't go or should not go?
I mean, my concern with the inevitability is I believe that that's probably going to happen just based on our history of technology development. My issue with that comment is it kind of just says we're not going to have a public discussion about it, right? That it's inevitable, and so you might as well just accept it and move on. And that is the part that I have issue with, because then we can't have the discussion I think you're asking is there then a hard stop? If the technology is inevitable, then there's probably is no hard stop if we sort of follow that logic. And what I would suggest is that we broaden this discussion so we actually hear from other people. I mean, the Oxitec case study is one I think we should really follow closely, because it's one where I think they did think it was inevitable. And now if you look at it, the public is in that area is very much against it. The regulatory system to evaluate that particular technology is, in my opinion, not the proper one. That FDA is the one that's evaluating it. So it adds to this notion that there's no one really in charge, no one sort of following on what's going on. And that doesn't help anyone. It doesn't help the issue you're trying to address, which is a serious one. They're trying to stave off the increase of dengue fever, which everyone believes will eventually get there. And the current methods don't work. They can only get about a 50% reduction based on the pesticide use that they're doing. But now that we've got the public sort of against it already, I don't really see how we move forward. But I think those are the things that we have to look at really seriously, particularly some of the ecological issues that right now there is no real funding that can address these things. And as we hear, the technology is already out there, so we're already behind in terms of evaluating these things, and we'll just be constantly playing catch up. And what I worry about is that we release these things without any sort of understanding of what's going on, and then we have to do a cleanup exercise, which is usually how the ecological impacts finally get addressed, is when there's a problem, not preemptive.
Do you think it's ever possible?
Maybe if I could just, I completely agree with that. If I could just sort of chime in. I think one of my favorite sayings is that we have a system that's perfectly designed to create the results it produces. And I think one of the first points I made was about the culture of science. I didn't say it disparagingly, but I think it's quite real. I think the notion that we better just kind of slow everything down and have a whole bunch of people that we're not really accustomed to talking to or to giving very much weight to their considerations. We better just start doing all that. I don't think that's really going to happen. I don't believe that we're going to have these, transformational changes in the culture. But I think that we are far enough along in our understanding of processes that can add value in terms of providing unique insights about what's at stake. And those need not derail science. They need not, stop it dead in its tracks. But I think if we do it well, they can actually reveal things that might be really important to pay attention to. Can help us do a better job. And in cases like the Oxitec Florida example, it may be that the interests amassed against a particular application really are too strong, and that's a decent example of where those processes may be working well. But that same process, if the engagement had been done differently, may very much have resulted in a different outcome.
If I might address that, I think that it is possible to change the culture of science. I think it is exceedingly difficult in many areas, but I think that if there is one where it could succeed, it is this one, because this is new. It is in effect, a new ecological niche for science that is currently being colonized. And the conditions for flourishing may be somewhat different, because we do have this additional imperative of community-guided research, because there are shared impacts in a way that is just not true of arguably any other field. I mean, there are certainly some parallels with, say, geoengineering, but again, the availability and low costs of this one really sets it apart. So I think that there is a chance that you could change the culture of science, in particular if you could work on changing the incentive structure, such that you could favor transparency and reward the disclosure of ideas and proposal stage research, if appropriately realized eventually, in order to encourage scientists to be transparent and engage all the way through. And I think that that is, especially through virtual platforms for engaging large numbers of people, could lead us towards a system wherein decisions are informed by the best scientific evidence, but ultimately determined by people's values.
Okay, Ken, quickly, if you would.
If you look to what's required to actually proceed with work in this area, I think the medical analogy that we've heard so much about is taking us down the wrong road. And the reason why is that if you're talking about a classic clinical trial and patient consent, the patient receiving the potential benefit is also bearing the risk. There are broader public benefits resulting from the knowledge generated. If you look at the examples that we've been talking about, that correspondence of individual acceptance of risk and potential acceptance of benefit does not coincide, which creates enormous potential difficulties for your committee as you try to figure out appropriate governance mechanisms.
Great. Joyce Tate, please.
Yeah, and my comments follow rather nicely from that last bit of discussion. They were stimulated by Jim Lavery's talk, but I think they're relevant to everybody. And thank you all for some really stimulating and very interesting background discussions that I think are going to help us a lot. When Jim was talking about the community engagement around a disease-related issue, it struck me that you could, in fact, find a community in whose interests this particular technology development was, who would, if they were acting rationally, be very favourable towards a particular development. But that development, as Ken was saying, might actually have serious ecological consequences downstream, which would not be in the interests of the broader community. And so you might end up wanting to say, no, we won't go there. Having engaged with people in advance about their desire for the technology, and then you say, no, we can't do it. That's something to consider when you're doing your engagement. But I also thought the kind of community engagement that Jim Lavery talked about was, if I may say so, the easy end of the engagement process, and where you are dealing with a smallish community in whose interests this particular technology might be. It gets much more difficult when you're talking to broader societal groups, about broader issues like the whole science of gene drives and CRISPR and Cas9 and the issues around whether that scientific research itself should proceed or not, and where you get people engaging on the basis of values and ideology, where you're in danger of actually leading to polarization by having the engagement if you're not very careful. That was very much what happened in Europe in the context of GM crop development. And I think these are issues that we will need to think about when we're doing our deliberations. And I just wondered if the speakers have got any thoughts on how we might resolve some of these issues.
I mean, I can speak to it in terms of what's happening at the UN CBD level, and I would say you're right, that it's complex is a nice way that I would put it, since we're on record here. And I don't see how we're gonna get through it. I mean, I sit on the ATAG with, for the CBD, which is evaluating synthetic biology now, and we're tasked with coming up with, in agreement, on a definition of synthetic biology, what the risk assessments of synthetic biology are going to be, what the impacts of synthetic biology are going to be, and that includes the societal impacts, the indigenous rights, the access and benefit sharing, and the differences in the communities based on where you're located, just on a country level, are vastly different in terms of what the interests are and what their immediate needs are. And so to come up with some sort of like universal system for that, I don't actually think that that's the proper way to go about it, because I do think that it's going to wind up being a case-by-case kind of evaluation of the particular application that's being used, and a case-by-case application in terms of the community where it's being utilized, even though it has this potential to spread. But I think that's probably where we have to start, because trying to get a consensus in an international context, in my opinion, is near to impossible.
Ken, did you want to?
I'd recommend going very narrowly for as beneficial and narrow an application as possible, where you do have a community that's benefiting substantially. You may actually want to be going with a community like Lyme, Connecticut, instead of a developing country where accusations of colonialism may exist, and where there's no easy substitute. I'm not sure. But it comes down to particulars, as Todd said. To be talking in generalities about gene drives or about synthetic biology is certainly a discussion that has to take place, but it's going to come down to particulars with specific risk-benefit calculations and benefits, and significant variations in degrees of uncertainty.
Jim Lavery, did you want to come in?
Yeah, I mean, just a couple of thoughts. I mean, one is that, thinking about Tony's experience in Mexico, one of the things that was very clear, and this is reflected in Laura's PhD thesis that I'll send along, and in the papers that are on their way, is that the community saw the potential benefits of the research in a much broader sense than just the gains that would be delivered through the science. They saw them in political terms. It would put their communities on the map. It would do all these other things that were ancillary and complementary, if you like, to the science. So that's one thing to keep in mind. I mean, the second is probably an unresolved question that I didn't have in my list as well, and that's the relationship between community engagement and public engagement. And part of that may just have to do with scale, but I think that part of it is the so there's a lot of really good work actually done on mechanisms of public engagement around technology assessment and the introduction of new technologies, and we're very mindful of that work in our own work on community engagement. But if you think of, say, privacy as an example, if when we come up with principles of either in the law or in ethical guidance around how privacy ought to be maintained, say in communications. As our communications networks proliferate and become gargantuan, we have to try still to preserve those kind of fundamental principles about privacy and protection as we scale. So what we're interested in is how then do you identify the core ethical ideas that need to be kind of captured however wherever you go through the scale, and see if you can build with some of these tools in public engagement to try to preserve those kind of ethical dimensions as you scale. So I'm not sure that there are any easy off-the-shelf methods, but that's really part of what our work's going to involve going forward as well.
All right, a comment from Lita Nestetu, and then Kevin, and then I have two questions from the committee, and we'll wrap up this session.
So I was thinking as all these comments were being deliberated that I think it's important to remember where this discussion is taking place. So this is a discussion about a technology that is developed in very rich countries, and where eventually the benefits or the outcomes of this technology will be sought after by other parts of the world. The GMO analogy is very interesting because Europe had the financial ability to, let's say no to them being used, where other parts of the world do not have that luxury. And that's where issues of justice come in, and that's where the human subjects analogy is relevant in the sense that it is an internationally understood framework for how you're supposed to be doing this kind of research. It doesn't mean that it applies here one by one, but it is something where it is recognized in all parts of the world, whereas other frameworks are not. Laboratory animal frameworks are not, misuse frameworks are not. The human subject is the only thing where everybody gets why it's important. So the issues of, I don't know, equity, equipoise, justice, however different kinds of words need to apply here, I think they are important because this is a technology that has the potential to change everything. And, people, it's developed here, but all the ramifications will eventually have to be used elsewhere, and how you bring the scientists of these countries who may not be educated in these technologies, but who have nevertheless the connections to the communities in those countries is another issue that is very important, and how they understand this, how they feel part of the scientific community that sort of engages them and plays in the same rules with them is a very big, I don't know, stepping stone, a block or something, whatever you want to call it. So these are just some thoughts.
Kevin.
I'd just like to make the brief point that there is a tremendous difference between community engagement at the point where the technology has already been developed and has become a product, and now they are seeking entrance into a market, which has been the case for virtually every technology developed to date. This one is different. There is no role for the market, which removes even the semblance of consent through mass adoption. And we at least have an opportunity, because it is so early and we are addressing it so early, to have the community guide the development of the technology from the very beginning. We've been emphasizing over and over, this is going to have to be evaluated on a case-by-case basis. Let's have that evaluation and the engagement start at the very beginning when the scientist writes the grant proposal. That should be made public.
Okay.
The conversation should start then. Like you should bring in the scientists from other countries who would need to be the collaborators at that stage. You should invite people's input from the potentially affected communities, and in fact, everywhere at that stage, to help identify things that might go wrong, ways in which it might be made safer, and build public trust as early as possible.
So you're calling for what Dave Gustin and others have talked about in terms of anticipatory governance, basically
Yes.
getting in front of the technology.
But there's one very important point that we need to pin down. If you look at gene drive applications as distinct from many other applications of genetic engineering, the companies are less interested in it because you're talking about one-time changes that then pass through environments, and it's difficult to cash in. And that changes the economics of this application quite a bit compared with conventional biotechnology applications.
It also changes the IP interests.
Yes.
Right.
For the same reason, which also may enable transparency in this field where it is not an option in lots of other biomedical research.
Dr. Ache?
Yes, thank you. I have two questions, actually. One is for Kevin and one is for James and maybe Tony, if they're still on the line. Going back to the technology, if we want to monitor and survey where the drives that are positive are throughout the population, talk about sequencing, but that could be a challenge for some of the countries in which we're talking about maybe being this technology being deployed in. Is there exploration about markers, other kind of markers, indicators for having the guide RNAs so we can track them? That's the first question. Second question was to both you and James. You talk about polls that have been performed. In some regard, will the public accept this? Yes, no? Are any of those polls including specific comments from your target populations to their concerns that we should also be considering that maybe are not being brought up here? Do we have access to those survey results? Same with you, James, from Tony's studies?
All right, very briefly on the monitoring. Right now, the answer is no. You could certainly do a PCR-based assay to detect the most commonly used form of Cas9. So vast majority of gene drives would probably use that one. Certainly most unauthorized gene drives would use that one. And yes, so that one, you can in fact pick it up very easily via a PCR assay. Is anyone actively looking into this and the feasibility for environmental monitoring in that manner? No, that's one of those projects that, as Todd emphasizes, really ought to be funded and begin now. For the other ones, you really do need environmental metagenomic sequencing, which is an area of active research, but not for this particular application. I'm going to defer on the poll question to those who know.
That last poll that I showed, you can have, I can send that to the committee. We actually had it as an online one so we could enable people to actually write their concerns, and it was actually a secondary question of what are the positives you see in this, what are the negatives, what are your concerns? And so I'll send that to the committee and the full data if you want the binder full of it. But yeah, I'll provide that data for you.
Jim Lavery, anything else to add on that?
Yeah, not, certainly not related to any of the work that we've been involved in. I think that, there are, I think the polls that I've seen have been more kind of just general awareness and perceptions and support or not support kind of things. One of the problems with polls and surveys is that they are, by definition, they are framed from the perspective of the people asking the questions. And so one of the points in the ethical aims of community engagement that we differentiate is this notion of these activities helping to unearth what we think of as non-obvious factors. And so if you have a question in a poll or a question in a survey, by definition, they've become obvious to you. And so I think it's an area, though, where we can probably do a lot of work in trying to figure out from the insights that we've gained, say, in our case studies, build better instruments, polling, surveys that can get deeper into some of these things that have been unearthed as maybe non-obvious when we started, but now are been brought to bear. But no, I actually don't know of too many polls explicitly in this area.
Okay, thank you. We're going to go to Wayne Landis for the last question before we take a quick break, and then we go to the public comment. So we have to stay on time as far as the public comment is concerned, since we'll have individuals coming in.
I heard
So
the
Wayne-
hint. I'll keep it short.
Okay.
Community engagement. The examples I heard were all very much directed towards health. So what about the extensive literature and experience in community engagement with the cleanup of contaminated sites, remediation and so forth? I didn't hear any of that. We have a huge amount of experience, much of which I think is applicable. Was that just an oversight, or because you're focused so much on particular case studies, or was any of that information actually looked at?
Yeah, well, so 15 minutes is not time for a course. One of the things that we did early on is we did, and this is one of the things that's in the pipeline that we'll hopefully be able to get out in the next 6 months or so, but was to try to just do a broad literature review. One of the problems in this area is that there are literally thousands of papers published in this kind of space about community engagement, public engagement. There's a million different terms. But we tried to kind of look at this. There's all kinds of stuff in environmental areas and forestry, and across the gamut. What we were looking for specifically was some explicit attention to what it meant to claim that these activities had been effective, whatever that might mean. And what we found was surprisingly little explicit attention on that kind of evaluative side of these activities. There's lots of excellent description. There's lots of great cases. There's no lack of that. But the papers and examples that cross over into providing insights about how we might think about the effectiveness were far fewer than we expected. So it's an area that we're, in the next 2 years in my own work, I'm going to be going back through some of that literature and trying to pull out some of those lessons. But yeah, it was an interesting finding.
Yeah, I have one more. I'm not sure it's a question or comment. I can think of species hybridization happens all the time. Bent grasses exchange genes. One of the best cases of GMO genetic transfers, bent grasses in Oregon. And that would certainly be a way of transmitting this trait. Fish hybridize all the time. I mean, species hybridize all the time. I see no reason why these genes would not move into those species as well. And yeah, so I'm trying to understand what those rates were going to be, how easy it would be to detect, and this containment thing. Be careful with islands. Did you guys read the book Jurassic Park? The dinosaurs made it off the island. All right.
So,
Okay, we'll go and finish up here quickly before we get totally into Jurassic Park, but go ahead.
So on the issue of hybridization, it would depend on how different is the target sequence in the species with which you're hybridizing. And in terms of how likely is it to cut that sequence or to evolve to cut that sequence, that's the sort of thing where I think we need the populations of model organisms where we can play with the mutation rate, where we can grow billions of organisms side by side representing the target population and the related population in order to measure whether or not that's going to happen.
Note that Kevin is not assuming stability, and many people operating in the field are. And this is no, I know, I'm not indicating you are. But the point being that the research agenda that we're talking about is really important because we don't understand long-term properties of a lot of these things.
All right, with that, we're going to break until 4 o'clock with a firm start at 4 o'clock because we have public comments coming in. But let me thank the committee, the panel, sorry, the individuals who are here, as well as Jim Lavery virtually, for a really informative set of presentations. They will be very helpful to this committee. Thanks very much. So we'll break for just about 8 minutes till 4 o'clock, and then we'll start in with the public comment section.